now I'm going to extract some pigments, the photosynthetic pigments from leaf tissue. So for this experiment, we're going to be using a couple different types of solvents to dissolve and extract the pigment. Now, if you remember from your investigation of photosynthesis, the pigments, chlorophyll and carotenoids, they're in the membranes. Okay, and membranes are phospholipids. They're lipids. They're fat. And what we remember about lipids is that they're non-polar. In other words, they don't dissolve in polar things or polar compounds like water, for example. Water is polar. We all know oil and water don't mix. Well, that's because water is polar and lipids, fats, are non-polar. Well, molecules can be non-polar also, and in fact pigments like chlorophyll and carotenoid are also non-polar. So they're actually fat soluble or soluble in non-polar solvents. So water is the universal solvent, as it's called, but it's polar. Now, some solvents can dissolve. You could dilute it with water, like alcohol. You could dilute with water. Another type of strong solvent that still can be diluted with water is acetone. But acetone is also able to dissolve fats, so it can kind of go both ways. It's called, well, we won't worry about that. So. Acetone, it can be used as a paint thinner, maybe not so much paint thinner. The fingernail polish removers often have acetone in it. It's a pretty strong solvent. Okay, if you leave it in certain types of plastic long enough, it'll actually discolor. It'll begin to kind of weaken and dissolve and attack the plastic. So we're going to start off with an extraction of leaf tissue with acetone. But because leaf tissue is full of water, just like all animal cells, plant cells, there's a lot of water there. And because water will mix with acetone, there'll be a lot of water still in our pigment extract. So we'll have to carry out a secondary step to get rid of all that extra water. And for that, we're going to be using a very strong non-polar solvent called hexane. Hexane will not dissolve in water whatsoever. It's completely non-polar. And it's a much stronger solvent. It will attack plastic. It'll dissolve styrofoam. So it's a very strong solvent. We'll use hexane in the final step to help us remove all the traces of water in our extract. So I've got some in this highly sophisticated scientific grinder here. It's just a basic kitchen grinder. And for our leaf tissue, we're just going to use some spinach leaves. So I'll just throw some spinach leaves in there. Obviously they're green, right? So definitely have a lot of chlorophyll in it. But also remember in our discussions that the other pigments are also here. So there are carotenoids and some other pigments. Within this leaf tissue, you just can't see them because there's so much chlorophyll in it. So this is the acetone. I'm just now going to just squirt some, splash some acetone in there. Yeah, that looks about right. Helps to mince up the tissue a little bit, so we'll help it out a little bit, mince the tissue. Okay. Okay, so here we have our green spinach smoothie. Although I don't think I'd drink this because the acetone would be poisonous. So don't want to drink this, but you can see now we've got the spinach leaves all ground up. Next we're going to use a very highly sophisticated advanced type of filter called a coffee filter. Okay, so we'll just stick the coffee filter down in here, and we're just going to filter out the chunks with this coffee filter. Okay, we'll just let that filter on through. So basically what we have here is what we would call a crude extract. We've ground up the tissue, so all of the cell membranes and cytoplasm and organelles, everything's been released into the acetone. All the membranes will have been dissolved, and fats and lipids in the cells will have been dissolved, or at least partially dissolved by the acetone. Acetone is also a very strong protein denaturant. So remember when we learned about proteins and how they can be denatured. So the proteins will denature. That makes them come out of solution and will initially make it look a little bit, maybe a little bit cloudy. But as this is filtering, so that's pretty good. That's good enough for now. There's still a little bit of chlorophyll coming through here, but that's more than enough for what we'll need. So I'm going to set this aside. And there is our chlorophyll extract. It's a very strong, dark chlorophyll extract. Hopefully you can see the definite green color in this extract. So even at this step, we could probably use this for spectrophotometry even at this step. Okay. But for the thin layer chromatography experiment, we really need to make sure we've got all the water out of the extract. So in this step, we need to carry out the next round of purification procedures where we follow it up with a hexane extraction. Now before we do that, we want to get rid of any precipitates, any proteins and DNA and carbohydrates that are left in there that will precipitate with the acetone. That means they'll come out of solution. So to do that, I'm going to transfer my extract to some of these tubes. We want to make sure these volumes are the same so they balance. So what a centrifuge does, this is a centrifuge here, and what it does is just spins our samples really, really fast. Okay, so the Earth generates 1 g of gravity, right? So that's the force of gravity. We always compare it to the Earth's gravity. So Earth's gravity is 1 g. Well, if you spin a sample in a centrifuge, these centrifuges can spin it up to thousands of RPMs. And some go tens of thousands or even hundreds of thousands of RPMs and generate many times more than just 1 g, the force of gravity. So in this centrifuge, so you can see, this is called the rotor, and it just spins. So I'm going to put these samples in the rotor. Close the lid, and then we'll run the centrifuge for about 5 minutes. Now this particular centrifuge, when it runs at top speed, it's generating over 4,000 times the force of gravity. So any solids that have formed, so like precipitating proteins, will be forced to the bottom of the tube, and in fact it'll actually form a very hard pellet in the bottom of the tube. This is a clarification step. This helps get rid of any cloudiness and sediment that's in our extract due to the precipitation of proteins and DNA and carbohydrates. So we'll let that go for about 5 minutes. Okay, let's see what we got in the oven. Okay, so this is our clarified pigment extract. So it's just a total leaf extract. I don't know how well if you could see it, but there's a kind of a whitish colored streak of material that has pelleted and formed a layer, has settled out along the side of this tube due to the centrifuge. Okay, so next I'll have to pour off the clarified extract, and then we'll be able to move on to the next step. Okay, so next I'm going to just carefully pour this extract into another tube, and I want to leave the sediment behind. So it should be pretty well tightly packed along the bottom of the tube. Don't want any of that extra sediment to come up, so I'm just being careful. There we go. So now maybe you can see this sediment that settled out on the bottom of the tube. So this is the proteins and DNA and other stuff from the cell that's not soluble in acetone. And now we've gotten rid of it. So our clarified extract now is really clear and ready for the next step. So let me go ahead and I can just work with these. So I'm going to divide this up again to 2. Okay, so this is a clarified crude extract of plant pigment from leaf tissue, but we want to go ahead and do the next step, because remember, this still has a lot of water in it. So we have to remove the excess water. To do that, I'm going to add some hexane. As I said, hexane is a very strong solvent. Does not dissolve in water. And so let me just pour some hexane in here. So you can see now where I've poured the hexane, can you see the light layer that's sitting on top? That's the hexane layer. The chlorophyll is already starting to dissolve in the hexane, because the chlorophyll would rather be in the hexane than where the water is with the acetone. So what I'm going to do is just start shaking this, and what we want to do is we want to extract that chlorophyll out of the aqueous layer, that's the layer with water, and transfer it to the non-aqueous layer, or the hexane layer. So nothing fancy here, just going to sit here and mix it for a while, and I'll show you what it looks like after I'm finished extracting it. Okay, so now I'm going to go ahead and give it another round in the centrifuge. What that will do is help separate the aqueous and non-aqueous layer. This step doesn't take very long at all. Probably long enough. Won't let me open it until it's completely come to a stop. There we go. Okay, so look now. So see what happens is now all the chlorophyll is up on the top layer. There's still a little bit of yellowish, kind of greenish color to the bottom, so we didn't quite get it all, but this is plenty for our purposes. So the chlorophyll has now separated and is dissolved in the upper layer where there's no more water. So we've got there's probably some acetone and hexane and the chlorophyll in this top layer. So the last step I have to do is to draw off this top layer, the green layer, and that is now our purified plant pigment extract. The final step is to just recover this top layer. So what I'll do is very carefully, using this pipette, I'm just going to draw this upper layer off. And what we want to be careful not to get any of this lower layer, because remember that's full of water. I'm not going to try to get every last drop of it, because if we get too close we might start sucking up some water. And you can see all of the tubes that were left behind. And now here we have our purified chlorophyll extract. Really it's a total leaf organic solvent extract. So it's probably got membranes, lipids dissolved in it. Any of the nonpolar compounds will be dissolved in it. And of course the pigments are dissolved in it. Still looks pretty green, but as I've said, there's other pigments are present in here that you just can't see because of all the chlorophyll. And in the thin layer chromatography procedure, we'll actually separate this mixture into its component pigments, and we'll get to see some of the other pigments that are present within this extract. Okay, so to carry out thin layer chromatography, we have to use these little strips of coated plastic. Okay, it's just on a plastic backing, but you can see the wider side kind of looks a little bit powdery. I don't know how well you can see it, but it's got a powdery coating. Okay, it's a silica, kind of like a very fine ground glass coating, which we'll apply our sample to. So here's our purified extract, and we don't need very much of this. So I'm just going to pour a little bit out into this beaker, and that should be more than enough. Okay, and what I'm going to do is I'm going to apply a line of pigment close to about 1 centimeter off of the bottom. So I'm going to mark a little spot there with pencil, not pen, but with pencil, and use these really fine capillary tubes to apply it. So you know how if you've ever been drinking a Coke or tea or whatever with a straw, and you can stick the straw in there and hold your finger on it, it'll hold the liquid up in there. So I'm just going to stick this down in there, and when I pick it up, you can see that the pigment stayed up in the tube. So carefully, I'm going to apply dot at a time. You can't drag it. Just put a dot, a row of dots going across. Okay, and then we'll wait a few seconds and then reapply it, and I'm going to do this repeatedly over and over and over again until I get a really dark line of pigment. Doesn't take too long to dry in between. Hexane is very volatile and just evaporates really fast. So you wait a few seconds, make sure it's completely dry. If you apply it too quickly, if it's still really wet, it'll spread out too much. So you want to make sure you let it dry in between. And you don't want to scrape. You want to drag the capillary tube along the coating, because it'll scrape it off and then that'll mess it up. And so I could probably really even get away with a finer capillary tube, because these are kind of spreading more than I want, but it'll be okay. So I'm just going to keep doing this repeatedly until I have a really dark row of dots. Okay, so now we're ready to separate the pigments. So to do that, we're going to drop this strip, the TLC strip, into this reservoir or jar that's got a little bit of the solvent in the bottom of it. It's a hexane acetone mixture. We keep the jar lid closed so the atmosphere in here gets completely saturated with that volatile solvent. And I'm going to very carefully check first. Want to make sure that the solvent is not going to cover our pigment. So the solvent should not come up over it because it'll just dissolve it and take it right off. So I'm just going to hold it up against it here real quick, and I think I've got too much. So I'm going to pour some of this off. It's higher in the middle, but I want to make sure that we don't dissolve our pigment off. And okay, so we'll try that. Just a little bit of solvent in the bottom. I think that should work. Maybe just a little bit more. Let's try that. So I'm going to carefully lower this strip down into the jar, and once I get it in there, I'm just going to leave it alone. So what's going to happen is the solvent now is diffusing through the powder coating, and once that solvent hits the pigment on there, it'll dissolve it, and the pigments will then begin to move along with that diffusing line of solvent. Now, because the different pigments have slightly different solubilities in the solvent and slightly different binding affinities, so they're binding to the silica at slightly different strengths. So some bind harder and stronger than others. So the stronger they bind, the slower they're going to migrate. So the ones that bind weaker, less strongly, will migrate more quickly through the strip. The ones that bind stronger will migrate more slowly through the strip. And then all together, the different pigments will separate out based upon this relative solubility in the solvent and the binding affinity for the silica matrix. Okay, so to carry out thin layer chromatography, we have to use these little strips of coated plastic. Okay, it's just on a plastic backing, but you can see the whiter side kind of looks a little bit powdery. I don't know how well you can see it, but it's got a powdery coating. Okay, it's a silica, kind of like a very fine ground glass coating, which we'll apply our sample to. So here's our purified extract, and we don't need very much of this. So I'm just going to pour a little bit out into this beaker, and that should be more than enough. Okay, and what I'm going to do is I'm going to apply a line of pigment close to about 1 centimeter off of the bottom. So I'm going to mark a little spot there with pencil, not pen, but with pencil, and use these really fine capillary tubes to apply it. So you know how if you've ever been drinking a Coke or tea or whatever with the straw, and you can stick the straw in there and hold your finger on it, it'll hold the liquid up in there. So I'm just going to stick this down in there, and when I pick it up, you can see that the pigment stayed up in the tube. So carefully, I'm going to apply dot at a time. You can't drag it. Just put a dot, a row of dots going across. Okay, and then we'll wait a few seconds and then reapply it. And I'm going to do this repeatedly over and over and over again until I get a really dark line of pigment. Doesn't take too long to dry in between. Hexane is very volatile and just evaporates really fast. So you wait a few seconds, make sure it's completely dry. If you apply it too quickly, if it's still really wet, it'll spread out too much. So you want to make sure you let it dry in between. And you don't want to scrape. You want to drag the capillary tube along the coating, because it'll scrape it off and then that'll mess it up. And so I could probably really even get away with a finer capillary tube, because these are kind of spreading more than I want, but it'll be okay. So I'm just going to keep doing this repeatedly until I have a really, really dark row of dots. Okay, so now we're ready to separate the pigments. So to do that, we're going to drop this strip, the TLC strip, into this reservoir or jar that's got a little bit of the solvent in the bottom of it. It's a hexane acetone mixture. We keep the jar lid closed so the atmosphere in here gets completely saturated with that volatile solvent. And I'm going to very carefully check first. Want to make sure that the solvent is not going to cover our pigment. So the solvent should not come up over it because it'll just dissolve it and take it right off. So I'm just going to hold it up against it here real quick, and I think I've got too much. So I'm going to pour some of this off. It's higher in the middle, but I'm not going to, I want to make sure that we don't dissolve our pigment off. And okay, so we'll try that. Just a little bit of solvent in the bottom. I think that should work. Maybe just a little bit more. Let's try that. So I'm going to carefully lower this strip down into the jar, and once I get it in there, I'm just going to leave it alone. So what's going to happen is the solvent now is diffusing through the powder coating, and once that solvent hits the pigment on there, it'll dissolve it, and the pigments will then begin to move along with that diffusing line of solvent. Now, because the different pigments have slightly different solubilities in the solvent and slightly different binding affinities, so they're binding to the silica at slightly different strengths. So some bind harder and stronger than others. So the stronger they bind, the slower they're going to migrate. So the ones that bind weaker, less strongly, will migrate more quickly through the strip. The ones that bind stronger will migrate more slowly through the strip. And then all together, the different pigments will separate out based upon this relative solubility in the solvent and the binding affinity for the silica matrix. I'm going to zoom in here and see already you can see two distinctive bands at least, a very definite yellow color. That's a carotenoid band that's already resolved. Remember this green, the green chlorophyll extract, that yellow, now you can see that there's a yellow carotenoid pigment dissolved in it as well. So you can definitely see how the pigment bands are resolving. Let's see if we have enough chlorophyll. There's usually, you can see two chlorophyll bands, the chlorophyll A and chlorophyll B. We'll allow that to continue and see if we can get those two bands to resolve. So we'll just let this go until the solvent reaches the very top of the strip. And when the solvent reaches the top of the strip, it can't go any further, and that's when we stop it. So we're just going to let this resolve. Doesn't usually take too long. May have to add some more solvent here in a minute. I'm going to add just a little bit more solvent because I think what's happening is the bump in the middle is going dry, so there's not enough solvent. Now, you're going to see in a minute, I should not be using this kind of pipette because it's made out of plastic, and acetone will dissolve this plastic pretty quickly. Now this is dangerous. It's kind of hard to do this without messing it up, but we'll see. Okay. So now you start to see the two chlorophyll bands. You can see some additional carotenoid bands. There's a couple of much more slowly migrating carotenoid bands along with the very rapidly moving carotenoid at the very top. So, but now you can see also the two green chlorophyll bands that have separated out, chlorophyll A and chlorophyll B. The upper one is the chlorophyll A, I'm pretty sure. I have to double check that, I'll let you know. But I believe that's chlorophyll A, and it has a more of a bluish green tint. And the chlorophyll B has the more of a darker olive tint to it. So we'll just let that continue to separate until it's finished. So what we'll do is we'll stop when the upper carotenoid band gets close to the top of the strip. Now there's actually a way to identify pigments based upon how fast they migrate through a strip like this. And it's called the RF factor. And what you do is you watch where the solvent, the clear solvent, is migrating, and when it gets close to the end, you stop and you mark where it went, and then you mark how far each of the pigment bands migrated, and you take the ratio of how far the pigment band migrated to how far the solvent migrated, and that gives you a fraction. The highest number you can have is 1 if the band migrates right along with the solvent. But as you can see, most of the bands are running much slower than the solvent. The solvent front right now is about right here. You probably can't see it on the camera, but it's running right up above where this yellow solvent band is. But we're getting really nice resolution now of the bands. And we'll just let that go a little longer just so we can get these bands resolved more clearly. But this is very nice, very sharp resolution of bands. You can clearly see multiple types of carotenoids here. There's two that are running more slowly. You got the two green bands, and then you have this more rapidly migrating chlorophyll band. Carotenoid band. Okay, so I think we can stop our experiment now and take this out of the solution. So I'm just going to place it on this paper real quickly, and the solvent front is right here. So I got to mark it really quickly because it's going to evaporate, and then the wet spot will evaporate and you won't be able to see it anymore, so you have to mark it. So this is a very good example, nice run of the various pigments found within a leaf extract. So when you just look at the leaf and look at the extract, it just looks green. But in reality, there are many pigments. There's really more than one green. There's chlorophyll A and chlorophyll B, and then we have these other pigments also present in the extract. The worksheet will give you some additional information on how to identify these pigments and how to calculate the RF factor. So now you can see that a leaf, in fact, does have many different pigments in it. In addition to the main green chlorophyll pigment, there's also these accessory pigments that are also involved in photosynthesis that are not normally visible. In the fall, however, when the green chlorophyll starts to die, you can begin to see the carotenoid colors come through. So this shows you thin layer chromatography. Now I've got one more thing I'm going to show you. Now this is what you see under regular room light. But pigments also, if you've ever worn a t-shirt, maybe a bright colored t-shirt under a black light, you know how it fluoresces really bright? Well, pigments can fluoresce also. So when regular light shines on the pigment and it absorbs the photons, what you see is reflected is the light that's not, wavelengths that are not absorbed. But if you shine ultraviolet light on there, what happens is it excites the electrons much higher, and when they relax down to their ground state, they give off a different wavelength of light. So depending upon the wavelength of light that you're shining on it, the color might look differently. So I'm going to place this on an ultraviolet light box, and you'll see different colors. Okay, so here we go. So this is looking at the same strip now, but under ultraviolet light. And notice that the pigment stripes look quite a bit darker. Let me go ahead and hold it up here. Well, that won't work. And let's see, you'll notice this band here that wasn't as visible under regular light shows up a little better under ultraviolet light. There we go. Now you can really see that even better. Notice this band above what was green before. Notice now these were yellow bands, the carotenoid bands, but they almost look green or dark gray under ultraviolet light. But this band here looks like a definite kind of a pinkish color, doesn't it? So you can see the different colors. They look different based upon the wavelength of light that's shining on them. And there again, just for comparison, this band here, you can barely see it, but under regular light, and even these bands, you notice, they're starting to fade because they get bleached. Now that there's no longer the chlorophyll protecting them, these bands are getting fainter because they're absorbing light, and it's causing the bands to bleach. Photo bleach, it's called. Photo bleaching. Okay, so now we can see the several different pigments present in a leaf extract from spinach in this case, and you can definitely see that there's multiple pigments found in the leaf. for the last part of this demonstration and overview of plant pigment analysis, I'm going to show you how to carry out spectrophotometry. So a spectrophotometer is an instrument that we can use to measure the amount of absorption of light of a pigment, of different wavelengths of light of a pigment. So for example, we've got this chlorophyll extract. Now you watched me prepare, and throughout the course of this video I've introduced several very important major techniques that are used in many aspects of biological, biochemical, molecular biology research. Very fundamental, basic techniques. So in this last part, we're going to do some spectrophotometry. Now, this is the sample specimen that I've prepared, and it's a pretty rich green color. So you can see it definitely has plenty of pigment, and we've seen the mixture of pigments that are present with the thin layer chromatography experiment. Now, to be able to measure the absorption using this spectrophotometer, we have to make sure that our solution is not too strong, because these instruments have a maximum range. So we can max it out and we won't be able to get any information from it. So we first have to determine the correct dilution or concentration of pigment that will be within the functioning range of the spectrophotometer. And I have really no way of knowing that other than to do a series of dilutions. So I'm going to demonstrate serial dilution today. So, and then we'll use our diluted samples to carry out spectrophotometry. So basically what I'm going to do is take some of this solution, this extract, and I'm going to dilute it. And I'm going to do a series of threefold dilution. So I'm going to dilute it 1 to 3 each time. So I'm going to take, I've already added 2 milliliters of acetone. So I'm going to use acetone to dilute it. And so here we go. We're just going to go ahead. I'm going to take 1 milliliter out of this and add it to 2 milliliters of acetone. This will be our first dilution, okay? And, okay, stir it up really good. But we still don't really know if this is diluted enough. So I'm going to take 1 milliliter out of this and add it to the next tube. So this will be another threefold dilution. So in other words, now we've done a total of, in this case, this is diluted 6 times. I'm going to do that again. So I'm going to take 1 milliliter out of this and add it to the next. So now we have a 9-fold dilution. And I think I'm going to stop there because I'm pretty sure that one of these dilutions will work. We'll find out. If it's too concentrated, then it will just max out the instrument and it won't work for our experiment. So I'm going to go ahead and turn on the spectrophotometer, and it takes a minute to warm up. Might help if I plug it in. Funny how that works. Okay, so it's coming on, and kind of has to boot up, sort of like a computer. Now this is this is really a fairly old spectrophotometer. It's still pretty high tech. It's digital, and it works good for our purposes. This particular spectrophotometer will actually measure down into the ultraviolet range as well, but we're not going to use the ultraviolet range today. This device right here easily could run over $10,000. So just to give you a little feel about the complexity and level of technology that we're talking about here. So it's running through its startup procedure. So when it's ready, we'll do our experiment. Now while it's warming up, makes some noises and stuff as it goes. While it's warming up, let me talk a little bit more about the procedure we have to do. One thing we have to do, just like with when you're weighing samples using a weigh boat, you had to subtract the weight of the weigh boat. When we were doing the catalase assay, you had to measure the air pocket in this graduated cylinder and subtract that from all your other measurements. And we're going to have to do something kind of similar to that here. We actually have to start off by measuring the absorption characteristics of the solvent alone. Because the solvent, even though it looks clear, it still has molecules in it, and it still might absorb, probably will absorb some wavelengths of light and interfere with our measurement of our pigments. So we have to be able to account for that. And fortunately, with these machines, they'll do it automatically. We call that a blank. We have to run the blank. So when it's ready to go, we'll stick the blank in there, and it will automatically go through the entire wavelength spectrum and measure how much the light absorption of just the solvent by itself is, and it will automatically subtract it. So that's the first step once it's ready to go. So I've got it ready to go, and we call these tubes cuvettes, and the cuvette now, this cuvette just has the plain solvent in it, acetone. I'm going to put this down in the little holder, and I'm pressing the button that says measure blank. So this is the step that's going to subtract out any absorption of light due to the solvent alone, so we don't have to worry about that. So as I explained in the photosynthesis video lecture on how spectrophotometry works, there's a light source, there's a bulb, and it will shine through a prism which splits the light into its different wavelengths. And then there are filters, and the filters will select a certain wavelength range so that only a certain wavelength is shown through the sample. And then there's a photodetector on the other side of the sample. So depending upon how much light hits that photodetector will determine how much of that wavelength of light is actually being absorbed by the specimen. 90%. Almost ready. Okay, just from my past experience, I'm gonna try this one first. I'm pretty sure definitely the first one will be too concentrated, and even maybe the second one. So now I'm going to remove our blank, okay, and insert our diluted specimen. So this is the 1:6 dilution, and then we'll let it rip. The noises you can hear every once in a while, this is perfect. The noises you hear every once in a while, that's actually the machine switching filters as it runs through the wavelength spectrum. Now this particular instrument measures every 10 nanometers. So it starts off at 450, I believe, 450 nanometers, and we'll go through 700 or something. I don't remember right offhand, but remember 450, 400, that's the blue end of the spectrum, the highest energy, lowest wavelength for light, and then on the opposite end of the spectrum, around 750 nanometers, that's the red wavelengths of light. So, okay, so we have our data. Okay, so all it's done is printed off a row of numbers. We've got wavelengths in nanometers on one side and the relative absorption of light at each of those wavelengths. Now when you come to lab, I will provide this data for you to plot your own absorption spectrum. So long, folks.